All right. Hello, everybody, and welcome to the 2021 WESTalks. This is the third term now that we've been doing this, and very excitingly, our numbers, we've reached a much wider audience this term, thanks to a lot of help from a bunch of our friends of WESTalks promoting this. So welcome, everybody. So the Water and Environment Student Talks brings together leaders in sustainable water and environmental research from around the globe, which we've seen in the chat box. Everyone's messaging in where they're coming from. And students interested in making a difference. So it's a collaboration. WESTalks is a collaboration between IC Impacts and the UBC Future Waters Group. So IC Impacts is a pan-Canadian, Canada-India Research Center of Excellence, which serves as a new model for international collaboration, co-developing research solutions to solve global challenges. And it is hosted here at UBC and was founded in 2013. Whereas UBC Future Waters is a new UBC Research Excellence Cluster on the future states of water. And we focus a lot on interdisciplinary research at the intersection of law, policy, and governance, and the applied and biophysical sciences. So the organizing committee, there's 6 of us. There's Abhishek, Lili, Fuhar, Jaskaran, Furia, and myself. Most of us are from UBC, and Jaskaran is now from excitingly joining the University of Guelph. All right, so here is the lineup. I hope in the email that was sent out to everybody, you were able to see the upcoming lineup, which is basically this. So I'll let you go through it later, but kind of the changes that we've had since last term, we found there was lots of interest both in the kind of technical, possibly you could call them maybe towards more engineering talks, as well as the WASH talks and the global development talks. So we tried to alternate weeks on that, and depending on speaker availability, sometimes it's not always alternating, but we tried our best to do that. And especially later in the term, we've actually booked speakers all the way up until June 3rd, I think. We get a lot more into that alternating weeks. So that's quite exciting. But today, it's my honor to now introduce, first, all the previous WESTalks are available on the IC Impacts YouTube, which also there was a link sent out in the email. And also in the email, we sent a calendar invite, so feel free to add that to your calendar. All right, and now it's my honor to introduce today's speaker, Dr. Lutgarde Raskin, who is one of the longest and most accomplished resumes I've been able to read, had the honor of reading. So she's the Vernon L. Snowyink Distinguished University Professor of Environmental Engineering and the Alteram ERIM Russell O'Neill Professor of Engineering at the University of Michigan. And she's been there since 2005. She serves as the Associate Dean for Academic Programs and Initiatives at the Rackham Graduate School. Before her University of Michigan appointment, she was a professor at the University of Illinois at Urbana-Champaign for 12 years. She received her bachelor's and master's degrees in bioscience, engineering, and economics from the University of Leuven in Belgium, and her PhD degrees in environmental engineering from the UIUC. She's a pioneer in molecular microbial ecology applied to water quality control and anaerobic bioprocesses. And she's globally recognized as an expert in microbial aspects of anaerobic waste treatment and drinking water treatment technologies. She's published about 150 peer-reviewed journal papers and about 400 conference proceedings. And Dr. Raskin is passionate about graduate education and has mentored approximately 20 postdocs and 100 graduate students, including 27 PhDs. She's an elected fellow of the American Academy of Microbiology and the International Water Association and the Water Environment Federation. And finally, she's an associate editor of the Environmental Science and Technology journal, which some of us may submit to from time to time, and we definitely all read. And was the Association of Environmental Engineering and Science Professors Distinguished Lecturer for 2018 and 2019. So with that, I'd like to turn it over to Dr. Raskin. I'll stop sharing. And so feel free throughout the presentation, if you have any questions, to message them in the chat box. And one of the other organizers will take over at the end, and we'll kind of in order of appearance of the questions, we'll have a question period at the end, and we're happy to stick around as long as Dr. Raskin is available and there's questions to answer. So yeah, feel free to just message those in the chat box. All right, take it away.
Thanks very much, Carl. Can everybody hear me, or Carl, can you hear me all right? The screen is good. Okay. Thank you so much for this really nice introduction and for inviting me. This is an exciting seminar series, so I'm honored to be here and to get you guys started this semester. I'm trying something new with my recent presentations in that I'm going to share with you the researcher utility partnership that we've been working on with one of the utilities here in Michigan, the City of Ann Arbor Drinking Water Utility. And the reason for doing this, rather than just presenting a technical talk, is that I've become more and more convinced that these types of partnerships can be really beneficial for both researchers and utilities and generally the water quality field. And so I'm hoping that I might inspire a few of you to start thinking about taking on beginning some of those partnerships yourselves. And I know some of you are probably already doing this. So I'm preaching to the choir for those of you who've started doing this already. I am going to start telling you a little bit of background before I dive into it. And this is because this is a general seminar series, I understand. And so some of you may not be that familiar with some of the things that we work on. Although this slide gives a very broad overview of the engineered water cycle. And I imagine that most of you are quite familiar with this, but this is just a reminder that everything is connected in the engineered water cycle. So we do drinking water treatment, distribute our water, we generate wastewater that is discharged back into the environment after treatment. And we have stormwater that's partially treated or not treated sometimes, ending up in the environment. And also we're more and more reusing our treated wastewater for drinking water production. With this slide, also want to emphasize that microbes are really important in every aspect of this engineered water cycle. And so we've started to use this term water infrastructure microbiome to emphasize that. So microbes are not just important in wastewater treatment as we've traditionally studied, but they're all over and are becoming more and more critical in treatment of drinking water as well. So water infrastructure microbiomes, when you start thinking about microbes in our engineered water cycle, you can't help to think about microbes in the natural environment because we have these close links between the natural environment and our drinking water systems. For example, we would draw water from surface waters, groundwaters, and so on to produce drinking water. So we naturally bring those natural water microbiomes into our treatment plants. And we also have natural connections with human microbiomes as we all consume water and so on. So I'm putting this up here to kind of state the obvious in some ways, but also to encourage you to develop those partnerships with colleagues in other disciplines, because we can really benefit from interactions with these other areas. And I feel pretty passionate about this. So I developed this kind of idea of the one water microbiome continuum. Everything in those different microbiomes is connected, and we should really be thinking about these interfaces between, for example, the natural water and the drinking water between and the human microbiome and so on. And if you are inspired to think more about this, you can take a look at this special issue of Current Opinion in Biotechnology that was co-edited by Per Nielsen from Denmark and myself, and where we talk about this idea a bit more, and where we've actually edited about 15 or so articles that really embrace that concept in different areas of the one water microbiome continuum. So that's a little advertisement for that special issue. The arrow that I missed here is the one that goes directly from the wastewater to the drinking water. And those of you thinking about direct water reuse for potable reuse, this arrow, of course, is really important to you. And microbes are a critical factor in making potable reuse a reality. I'm going to talk about researcher-utility partnerships in the drinking water field. So that is the top part of that schematic that I showed you before. And so the question is, can we really benefit and make a difference when we develop those partnerships? And I will try to answer that question through the rest of my talk, and you can judge for yourself whether we've made a difference. There's a number of benefits that I think can emerge from these partnerships, and this is how I'm organizing the rest of my talk today. So we are expanding our knowledge of real drinking water systems by developing those partnerships. We're making changes to real-world practices based on research results, so we can have a more direct impact on real-world practices by directly allowing implementations of our research results. We're also building capacity for taking on new collaborative projects. When those partnerships are in existence, it's a lot easier to develop those new collaborative projects. And finally, and really importantly, we are working together to co-create the future of the industry. And many of you probably know that we really have an issue with the workforce for the water industry. And so workforce development is certainly on our minds with these partnerships. And so I'm going to give you examples of all these 4 areas of things that we have worked on. And I hope you get inspired and you can imagine some of your own work fitting in any of these 4 areas. I'm going to spend most of my time talking about the first idea, and then I'll give you small examples of the other ideas towards the end of my talk. So before I dive into this, I know that not everybody thinks about drinking water all the time. So I want to give you a very quick overview of drinking water, maybe refresh your memory from an introductory environmental engineering course, or introduce it for the first time. What we do in drinking water is we make use of a multi-barrier approach to remove chemical and microbial contaminants. And we do that using a number of different processes. Coagulation, flocculation, filtration, disinfection are the typical processes that we have in conventional drinking water treatment. This is a schematic of a conventional drinking water treatment plant, and so you recognize these processes that I just mentioned. What I want to show you with this slide is that what I want to show you is that we focus so much on removing contaminants in the drinking water field, but it doesn't mean that we remove everything. In fact, in a typical finished drinking water that's perfectly safe to drink, you find about 1 million bacteria per liter of water. And in some other treatment schemes, an ozone biofiltration drinking water treatment plant, where you would have ozone treatment for primary disinfection followed by biofiltration before you add a secondary disinfectant. In that kind of a treatment scheme, you typically have even higher levels of bacteria in your finished water. Again, in most cases, perfectly fine to drink. This is just the natural drinking water microbiome that's present, but it's a relatively high number if you're not used to thinking about these kinds of things. I mentioned a few times that this water is perfectly safe to drink in most cases, but we are still concerned about some microbes, some populations of microorganisms that either may survive the treatment process, including the disinfection steps, or that may come into our distribution systems after treatment and potentially could even grow in our finished drinking water systems in the distribution system or in our home plumbing. And one of the populations of organisms we're concerned about are non-tuberculous mycobacteria. These are environmental microorganisms that we find in soils, we find them everywhere, and we typically find them in drinking water as well. And the reason we're concerned about these organisms is that some of them are opportunistic pathogens. That means that they're not causing disease in healthy populations of people, but they can cause disease in people who are immune compromised, very young, kids, elderly, and so on. There's a lot of different NTM, non-tuberculous mycobacteria. We know about 190 species. 20 of those species are opportunistic pathogens. And what we've observed, and when I say we, I mean the general research community, where this is a topic that's being studied by quite a number of people, that we've observed that there's an incidence of NTM infections. The incidence of NTM infections has been increasing in the United States, worldwide actually. And I'm giving you some good recent publications here in this slide. If you're interested in this, you could look up some of these papers to get a sense of the importance of these opportunistic pathogens. We find these in most of the tap water in the United States, and that's the same in Canada. 80% of the tap water that's been studied contains these NTM. We find them especially in shower biofilms. In fact, when you take samples from the biofilms in shower heads, you find that the community consists of about 10% NTM in some cases. And some of these NTM are clinically relevant. They fit within that group of 20 or so opportunistic pathogens. And so they have the potential to cause disease and cause infections. We've known about this for a while, but there's no drinking water standard for these NTM. Microbacterium avium, one of them, has been on the US EPA's contaminant candidate list for a long time. So it's been a concern, it's been on our radar, but there's a number of reasons why they haven't been regulated. I think there is a chance that they will become regulated, so it's certainly something that utilities are paying attention to. We started to study these NTM populations in our early work. This is a long time ago when I was still at the University of Illinois. I moved to Michigan in 2005, so you can do the math and see that this is a while ago when I started studying these organisms. We knew that they were fairly resistant to disinfectants. So for example, chloramine, chlorine, and ozone are typical disinfectants. And we did some pure culture work with Microbacterium avium, and we showed that when you expose them to low concentrations of the disinfectant chloramine, they develop an adaptive response, meaning that they become more resistant. And so that's a relevant observation for the drinking water field, because you can imagine that there is areas in the distribution system where you're exposing these organisms to low concentrations of chloramine. And so then they might become more resistant and persist in the system and start growing in your home plumbing, for example. This was work done in collaboration with Professor Marinas at the University of Illinois. I continued this work when I moved to Michigan with a PhD student called David Berry, and he studied the survival of these organisms inside of protozoa. So these organisms have developed a whole suite of mechanisms to be very resistant to disinfectants. This adaptive response is one mechanism, but they tend to also grow inside of protozoa, which makes them more resistant to disinfectants. They have other mechanisms that I'm not going to get into, but I just wanted to share a little bit about the complex physiology of these organisms that are relevant for us in the drinking water field. So you could do a lot of very fundamental work with these organisms and, develop your whole career around that and have, find a lot of really interesting things that we don't know yet at this moment. But we decided to study these organisms in microbial communities because we knew, we wanted to really reach out to the relevant, the things that are relevant for the drinking water field. This is an early study that we did in Michigan, where we studied microbial community changes during disinfection. At that time, we were working in laboratory bioreactors still, so not directly working with the plant. And this bar shows you the complexity of this community in the effluent from a biofilter in our lab. And you see different populations, relative abundance is the measure that I have here on the y-axis. So you see different populations showing up in this sample. When you expose this community to the disinfectant chloramine and you wait for a while, so you see a complete change in the life fraction of the community. So I'm plotting here only the organisms that are still alive when you expose them to chloramine. And in the x-axis, I'm plotting the concentration of chloramine multiplied by the time of exposure. And those of you working in disinfection know that concept as the CT concept. So you could see that with longer exposure, we get a complete change in the community composition of the active community composition. And so there is an indication that disinfection really has an impact in this early work that we did in the lab. So we wanted to go back, or we wanted to start working in real treatment systems. And we're very fortunate that the city of Ann Arbor has an ozone biofiltration plant. So you've seen this schematic before. This is actually the schematic of the city of Ann Arbor treatment plant. We have plenty of places in this system where the microbial community in the water is exposed to disinfectants and oxidants. So ozone is the first time that the organisms are exposed to it. Then before you enter the biofilter, there's a residual concentration of ozone. So they continue to be exposed before they enter the biofilter. The effluent from the biofilter has low concentrations of disinfectant left. We have backwashing of the filter with chloraminated water. So we add chloramine as a residual, as a secondary disinfectant, and that water is used for backwashing of the filter. So the community in the filter is continuously exposed to the chloramine. So you could see that we have lots of opportunities for oxidant exposure and for changes in the community structure throughout this treatment plant. In the distribution system, again, there's residual disinfectant present, so further opportunity for change, especially because you have the residence time now, so you have a long exposure to the disinfectant in the distribution system. And let's see. I'm going to switch to showing you specific data that we obtained working with this real water system now. So all of what I've given you so far was a little bit of background to help you understand the data that I'm going to share with you. So as I mentioned, we are doing all this work with the city of Ann Arbor, and since I should have put a map of the US for those of you in different countries, but if you zoom into the Midwest of the US, you find Michigan. And in the southeastern part of the state of Michigan, we have the city of Ann Arbor, where the University of Michigan is located. And so this is where we've been working. As I mentioned, we have an ozone biofiltration plant. So here's our schematic again. And I've introduced to you this idea that there's lots of exposure to oxidants, to disinfectants. So there is a good chance that we will be selecting for these non-tuberculous mycobacteria. And when we started studying this with molecular methods a while ago, we did indeed see that these NTM populations, in terms of relative abundance, became more abundant further into the treatment scheme. So we took samples after biofiltration at the beginning of the distribution system, the finished water, and then in the distribution system. And you see this blue bar here represents NTM, non-tuberculous mycobacteria. And so they're definitely more abundant in terms of relative abundance. Knowing that about 10 to the 8 bacterial cells are present at that point, you could see that even a relatively low, maybe 8% or so of the community consists of these NTM. That's a substantial fraction of the cells. So I'm going to show you what we decided to do here in this system to get a better handle on this and try to understand what might be going on in this system. And so what we decided to do was kind of make our way through the treatment process to study these NTM populations in the different places where we thought there was selection for these organisms potentially. So we started out by studying the ozone contactor. And one of the beautiful things of working in a full-scale plant is that you can really get graduate students excited about sampling. So here we have photos of two of our graduate students, Nadine Kotlars and Nicole Rauke. Nadine was leading this study. So Nadine and Nicole had an opportunity to go inside of an ozone contactor, which is very unusual because it's a dangerous place, and so you could see there was a lot of safety precautions. But by going in there, they made observations that we would have never been able to get at, and the operators would have never told us because they're so used to it that they didn't think it might be relevant for our study. So what Nadine observed here, Nadine Kotlars. So you see the first author of this publication is Nadine Kotlars. I forgot to add her name to that slide, the previous slide. What she observed inside of the ozone contactor was that, so I need to back up here. I need to tell you that the top part is a schematic of one of the ozone contactors. And you see this is your typical ozone contactor with these different chambers. The green indicates the sampling locations that we have access to. And the ozone is added in this chamber here. And so the concentration of ozone is highest here and then is lower down the road. And this is the water, the finished water coming out of the ozone contactor that goes into the biofilter. Nadine used flow cytometry as a tool to monitor the live cells. We call them membrane intact cells because of the method that's used. And what you see is that initially you have a very high concentration of intact cells, and then it goes down right away when ozone is added. As you would expect, ozone is a very strong oxidant and kills almost everything. But you see it doesn't go down all the way. In fact, we saw an increase that we were very surprised about throughout the ozone contactor. And then it leveled off a little bit further. So when Nadine and Nicole went into the ozone contactor, they were able to take samples in different places and started to analyze the microbial community. And what they found is that there were membrane intact NTM surviving ozonation. We had not expected that at the time. We knew that they were resistant to disinfectants, but not so much to ozone, but they were surviving. You could see that the absolute concentrations of NTM in the influent of the ozone contactor are given here at the left, and then they went down in numbers as you saw in the previous slide, but they were still substantial, 5,000 NTM per liter of sample. Nadine and Nicole were able to take biofilm samples from the walls of the contactor and from sludge that accumulated at the bottom of the contactor. And then I'm going to go through these slides to show you the relative abundance of the community in those biofilm samples. And in red are all NTM populations. And what you'll see is that the farther we go into the contactor and in these different locations, you find a relatively high level of NTM. And so we speculate that the biofilms that develop in this non-ideal environment, this somewhat dirty ozone contactor, that biofilm helps with the survival of these organisms. So we learned a lot about the real life situation by being able to get into these ozone contactors. We found things that we would have never expected and would have never observed if we had done laboratory experiments. So now the utility knows that this is a potential concern, and they are certainly a lot more careful about cleaning their ozone contactor and dealing with the sludge buildup and with the biofilm development on the walls of the contactor. So moving on to the next phase, biofiltration and secondary disinfection, because that's, of course, the next step in the treatment process, we started studying NTM with different tools. And so, for example, you see here that we targeted a specific gene to monitor the absolute concentration of NTM in the biofilter influent and in the biofilter effluent. And you can see that we knew from our ozone study that there was NTM in the effluent of the ozone contactor, but we now observed that there was about 100-fold increase in absolute abundance of NTM across the biofilter. And we speculated that backwashing was an important factor in this because, as I mentioned before, the backwash water that goes into the bottom of the biofilters to backwash them every 3 days or so contains a disinfectant, an oxidant, chloramine, because the plant is plumbed in a way that they have to take their backwash water after they've added the chloramine. So is chloramine contributing to that selective pressure was a question we wanted to know. And we decided we first needed to know whether chloramine was actually present at measurable concentrations in the biofilter during backwashing. So here's our team at the time. You can recognize Nadine again, but there's other people, Nicole, Jimmy, and Sarah contributed to this work. And they were sampling here on top of the filter during a backwash period. So I really love those pictures that kind of show you that the working at these full-scale treatment plants can be really enjoyable because you have students and postdocs working with a real system, and sometimes that can be really inspirational for people that they know they're working with things that are real and are making a difference. So we measured chloramine concentrations at the top of the filter. The team that you just saw took those samples, and we measured that chloramine was present at relatively low concentrations, but we had measurable concentrations. In the reservoir, the concentration is about 3 milligrams per liter as chlorine. At the top of the filter during backwashing, we find these what we call sublethal concentrations because they're low, but they're sufficient to allow the NTM to survive and perhaps to even select for those NTM because they have that resistance to disinfection, the adaptive response that I referred to before. So we also wanted to know, yes, we find these NTM, but are they relevant from a clinical perspective? Do we find relevant NTMs in these full-scale biofilters? So we developed a method to identify the different NTM species, and Sarah Haig led this work. So she developed a long sequencing method using PacBio sequencing. We focused on a different gene in this case, and we also developed a sampling technique. So we, at the right here, you see this core. In this photograph, you see the sampling core going into the filter. So you're looking down at the filter, and you could see the granular activated carbon inside of our core. And then we took those samples and we used the PacBio sequencing method that Sarah developed to characterize only the NTM in this case in those 3 samples that we took from those cores. And what you see in the data here is that the relative abundance of NTM, only in this case, is primarily dominated by Mycobacterium avium, one of those clinically relevant NTM species. You can see that there's quite a bit of diversity in terms of NTM, but certainly M. avium is the dominant population. So that told us that what we're working on is relevant and we should continue and learn more. We also wanted to see if those organisms were active, what they were doing, because a lot of our work is based on DNA and doesn't necessarily indicate that organisms are active. So we developed a method to look at the transcriptional response of a relevant gene. These NTM have genes that are called mammalian cell entry genes that are interesting for a number of reasons. One, it it's a gene that's relevant for human infection, as the name implies, but it's also a gene that's relevant for entry into those protozoa that I had mentioned before. So we thought this would be an interesting gene to target, and especially if we would be able to develop a transcriptional method, we would know that that gene was being transcribed, which would indicate that these organisms would be active in our system. So we took those samples from filter backwashing, and we developed a method to monitor the relative expression, in this case, of this mammalian cell entry gene during the backwashing. And in the left y-axis, you see this relative expression in blue going up during the backwashing. On the right, you see again the chloramine concentration, that sublethal concentration of chloramine at the top of our filters. And that is going up in parallel with the relative expression of this gene during the backwashing time. So this suggested to us that yes, these organisms are clinically relevant, they're active, they're expressing genes that are relevant from both a human infection perspective as well as a protozoa infection perspective. So there's obviously a lot more to learn about these organisms in these complex systems, in these complex microbial communities. And we're continuing some of this work, but I wanted to show you what we learned about the distribution system and the home plumbing. So Sarah Haig led this work. So she sampled in quite a number of locations, and I'm just giving you a summary of some of her results here. I'm giving you averages of samples collected close to the plant in the distribution system, far away from the plant in the distribution system, so much longer residence time, and then inside people's home. And on the right y-axis, we'll start with the right y-axis first, you see total bacterial numbers are going up slightly when we move further into the distribution system and into home plumbing. So increased bacterial abundance, that's been observed before, not surprising. It's nice to see that you can actually determine these absolute concentrations of these organisms in these different samples. But on the left, we certainly have increased sample from the distribution system and in people's plumbing. So as engineers, we always want to correct problems, right? And so we're now studying what we can possibly do about this. Can we, for example, by reducing backwashing frequency, reduce the selective pressure for NTM? And this work was started a while ago. We did a full-scale experiment with 6 filters. So again, the beauty of working with these plants, you can talk the operators into doing full-scale experiments with you. So that's really cool. So we started with 6 filters and sampled them for a while to have baseline data. And then over time, we split them up into 2 groups. Our control filters continue to be operated as they normally were, with a backwash every 3 days. And then three of the filters were changed in operation in that the operators agreed to backwash them only every 9 days. So we simulated the reduced chloramine exposure by having less frequent backwashes in this experiment. And our data are quite messy and we're still analyzing them, but I'm giving you kind of a preview of some of the data that are coming out. You see the 3-day backwash filters on the left, the 9-day backwash filters on the right, and you see relative abundance of NTM in these plots here. What you see kind of as a general picture is that there is perhaps an indication that the relative abundance of NTMs in the filters is a bit higher in these filters that are more frequently exposed to chloramine than in the 9-day runs. But what we observed, and this is why I can't dive into this, is that there were other complexities that we hadn't been aware of in this full-scale system. And so we have a number of different factors that came into the study that we are studying. Those of you who know something about biofiltration or filtration in general know that there is, for example, underdrains in those filters. And what we learned is that not all the filters have the same underdrains. And that factor is actually important in the shaping of the microbial community in these filters. And so we had an unexpected complexity that we're trying to sort out. So the data are not particularly conclusive at this time because of these complexities. But this is kind of a preview of the data that we are working on at the moment to try to come up with strategies to improve this issue of NTM in our water. We're also studying this in a pilot scale study that we were able to set up with the utility after we wrote a proposal together to the Water Research Foundation. So we have this project in which we designed a study to do exactly what we did at the full scale level in a pilot scale scenario. And here we had a little more control. We were able to use backwash water that didn't have chloramine, in one case by using filter effluent, and in another case by using dechloraminated finished water. And that, again, the data are being processed at the moment. So I don't have time, and I don't really have them analyzed quite yet to show you. So I'm wrapping up this part of my presentation by telling you that these ongoing partnerships have produced a lot of knowledge about NTM in real systems. And we hope that this information is valuable beyond Ann Arbor. We know it's been really helpful already for the city of Ann Arbor, but hopefully by doing this full-scale work, other utilities can learn with us and perhaps get inspired to do some of their own work to investigate these issues. I mentioned that I would briefly touch on these other three topics. So making changes to real-world practice based on research results. The example that I wanted to show you is a snapshot of some of the data that we're collecting as part of a study focused on flushing protocols. The city needs to do compliance monitoring all the time. So they have this protocol that they use that's not very well developed based on our scientific knowledge. And so we wanted to investigate whether their protocol could be modified to make it more relevant for compliance sampling. And the data that I wanted to show you are total chlorine data that we measured at the treatment plant in the reservoir before distribution and at 4 different sites in the distribution system. The green data are the samples collected after overnight stagnation, the first draw of the sample after overnight stagnation. The blue data are the compliance monitoring data. So this is what the city collects all the time. And then we also collected the full flush, which we call ideal compliance monitoring, because that's the water that they should be monitoring from a regulatory perspective, the water that's in the distribution system. And what you see here, in some of these sites, there's huge differences in the chlorine levels in these different samples over time. So you could see we sampled here from January through October about, and you could see when the pandemic hit, the differences became more substantial in some of these sites. And so the first draw sample, for example, in site 2, the chlorine levels went down to 0 for some of these data points, even though the distribution system samples, the purple ones are totally fine. And so this isn't surprising, because some of the buildings where these effects were really clear were buildings that were closed during the pandemic. And the buildings that remained open had more reasonable chlorine data. There were still differences between the 3 different samples that we collected, but the differences weren't that substantial. And so this helped the utility in a couple of ways. They were recommending flushing programs to these buildings that were closed. So they had some detailed data to help inform the flushing protocols. But in the future, it's also going to help them think about their own flushing protocol for their compliance monitoring sampling. So that's the example I wanted to show you, that having PhD students do work with utilities can really have an impact on work that the utility is doing on a routine basis. The third point, building capacity to take on new collaborative projects, is an exciting one. This project that I briefly want to tell you about came about because the utility was changing their filtration strategy because they had problems with PFAS. PFAS has become a big problem, and I've seen some of your seminars coming up are actually going to talk about it. We've had a lot of PFAS issues in Michigan, and the city of Ann Arbor is no exception. So the strategy to deal with the PFAS in the source water, since there was no quick way to remove the PFAS from the source water, was to improve the filtration. And the cities decided to switch from a particular carbon that they used, which is called F300 GAC, to a new carbon that's better in absorbing PFAS. They also decided to change the frequency in which they replace the carbon. So from their 5-year change out, they are now going to a 2-year change out. And so we were studying their filters. And so we decided to look at this as well, because whenever you add new media to your filter, you have a startup issue when you have a biological community that needs to develop on these filters. So we monitored filter startup, and in particular, we were asked to look at nitrification, because what the utility noticed is that during startup of those new filters, they were starting to see higher levels of nitrite than they had previously observed. And so certainly the startup may be related to this. And I'll show you just some brief microbial data. This is a PCOA plot, a principal coordinate analysis, where each dot represents a microbial community. And if I add some ovals here, it will help you to see what's going on. We took samples from the old filters and plotted them on this plot. So the microbial community in lots of different old filters that had GAC ages between 1 and 6 years are all quite similar. But when we switched to using the new GAC, the F400 carbon, initially the community in the filters was very similar, and that's because we used some of the carbon to seed those filters, but then they developed a little differently over time. So the community deviated from that established community after 1 month of operation, and then gradually, after 6 months of operation, moved back to the community structure that we had in that original inoculum. So we are now continuing this study with more sampling to study the startup in more detail. This is just a snapshot that we were able to get from that other study that I mentioned before. So now we're studying nitrification in more detail and hopefully provide some guidance for them to avoid the buildup of nitrite during these startup periods. And then finally, I wanted to just briefly tell you what we've been working on in terms of workforce development and working, co-creating the future of the industry. We decided to organize a workshop, which we call the Drinking Water Innovation Salon, to inspire other utilities in Michigan to develop partnerships. So we invited about 25 utility leaders and 25 academic researchers from the 3 main universities in Michigan, and we organized this this workshop. And it was a really inspirational event for a lot of people, and so we're hoping that efforts like this can inspire other researchers and utilities to work together. We are doing a few other things, and one of those is that we have a really wonderful water treatment manager in the city of Ann Arbor, Brian Steglitz, and he is teaching one of our graduate courses, and it's called Practical Engineering Solutions to Drinking Water Challenges. And just yesterday, I reviewed his course material from last fall, and I was looking at the student feedback, and it was amazing how well the students liked his course and how they were indeed inspired. And some of them said, well, you know, this has opened my eyes. I would love to work in a utility. So Brian was really happy with that feedback. We are, you know, providing these real hands-on real-world learning experiences for our students that they otherwise wouldn't get when faculty teach their courses. And of course, this is beneficial for the industry because now these students are inspired to potentially stay with the water industry. So this is kind of a summary slide with some of the photos that you've seen before and some new ones. I could tell you many more stories, but you know, I have only so much time. So I wanted to end with this encouragement that, you know, we found a lot of really relevant information, innovative information, cutting edge research that we can publish in high-end journals. But perhaps it's more meaningful than doing some lab work. And the students generally find it a lot of fun, not just the students. I get really excited about some of the work as myself. And I want to end with acknowledgements. And you could see here that the people at the treatment plant are high up on my list. Brian Steglitz, who I mentioned already, Sarah Page, who is the water quality manager, and then a lot of the operators and other people involved from the city of Ann Arbor treatment plant. And here's our pilot study team in front of our pilots. And then the second acknowledgement slide is really the current and former students and collaborators. At the top, you see, some of you may recognize some of the former students and postdocs that I mentioned, or that you've gotten to know personally. The second row, again, is people that were involved more recently in our work. The third row is current postdocs and graduate students. And then finally, at the bottom, these are faculty collaborators from the University of Michigan. And we have people from civil and environmental engineering, from the School of Public Health, and from the medical school. We have a few clinicians, John Lacuma and Lindsay Caverly. They're clinical infectious disease researchers who study organisms like NTM and have been really instrumental in helping us think about these issues. And I'll stop here, and hopefully there's a little bit of time for questions.